everyone, and welcome to today's webinar titled Concept-to-Commercialization, Thermo Fisher's Unique Approach to Rapidly Advancing Your Antibody and Protein Therapeutics. This webinar is a part of the 10th Annual Genetics Virtual Week. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information about Thermo Fisher Scientific, go to thermofisher.com. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find the live chat located at the right of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. In addition to asking questions, we encourage you to participate in 3 poll questions in the middle of the speaker's presentation. Click on your answer when the poll questions are displayed. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. I now present today's speakers. Steve Lewis, Senior Product Manager, Gene Synthesis and Synthetic Biology, Thermo Fisher Scientific, and Jeff Howe, Ph.D., Director, Scientific and Technical Affairs, Pharma Services, Thermo Fisher Scientific. For a complete biography on our speakers, please visit the Biography tab at the left of your screen. Steve and Jeff, you may now begin your presentation.
Great. Thank you, Maria. I really appreciate you hosting us for today. I will be doing the first half of the presentation, and I'm very excited to be here for today. First, I'd like to talk a little bit just about Thermo Fisher in general. We are, of course, a very large organization, but hopefully today through this webinar, we'll help with navigating it and really seeing how small and integrated our company truly is. We take a lot of pride holistically across the company in our mission to enable our customers to make the world healthier, cleaner, and safer. And today, for our presentation, we're going to move into talking about the agenda. The first half will be primarily about services provided within Thermo Fisher Scientific for early stage discovery, in particular antibodies. And we will talk about why starting with the gene or gene synthesis is the fastest way to move ultimately into the clinic from R&D into commercial scale production. So with the agenda here, we're going to start really at the beginning of the therapeutic journey and partnering with us. We're going to talk about optimization of your gene, as well as very advanced next generation gene synthesis, as well as advanced protein expression techniques. And then also how you move from going from selecting your candidates from R&D or in the R&D environment to ultimately production. From there, my partner Jeff will be speaking about strategies for accelerating your timeline to clinic, mitigating risk, as well as building a foundation for success and how to get started with Thermo Fisher right after this presentation. So to provide a little bit of context, Thermo Fisher Scientific has been working in the infectious diseases world for quite a long time. And we wanted to start here in terms of providing context for today's presentation because we have a long history working within infectious disease. Starting in 2000, we had a very early infectious disease response effort that we worked on in terms of HIV, all the way through the mid-2000s and all the way to today. And of course, you can read the whole timeline, and if you're watching this as a recorded presentation, you can pause it and take a look at all of the different details that we've been working on for more than 20 years in this space. But I'd like to highlight 2020 and 2021, where we really did have an impact, a significant impact, partnering with many companies around the world in terms of our response to SARS-CoV-2. This was a very rapid and very serious and very interesting time, not just for us in the biotech space, but really globally. And it was very challenging and also rewarding to work on a lot of these early stage activities in terms of pandemic response. And so if you have any questions about some of our activities in that space or how we contributed to the development of some of the vaccines and therapeutics as an organization, we'd be happy to talk about that. We also have some materials that we'd be happy to send after this presentation to explain a bit more in depth related to how we've worked toward developing critical vaccines at unprecedented velocities. So for today, we're going to start really talking about the early steps of therapeutics and vaccine development, starting with the early stage discovery and research through development, clinical trials, and then ultimately commercial scale production. As I mentioned earlier, Thermo Fisher, of course, is a large organization, but today, our focus for our webinar is really to show how you can go from idea all the way through commercial production in a relatively seamless way, working with our groups in Thermo Fisher Scientific. And for the first part of today, we are going to focus on the early discovery and research aspect as well as development before moving into the clinical trials aspect as well as commercial production with our partner, Jeff. So today, starting in the early discovery and research, we really focus on the idea of using recombinant DNA based just on sequence information at the very beginning to optimize your protein production. Within our production organization, we do have the ability to rapidly create DNA from a de novo gene synthesis processes, but believe it or not, we actually start in a digital way. We start with a proprietary algorithm that really helps us to optimize the gene expression or the protein expression resulting from your gene synthesis. And so I'll talk about some of those services today, but in particular, I'd like to start by talking about the discovery workflow. What you're seeing on the screen is what we believe to be a very reasonable timeline for starting at the R&D scale, or producing what we refer to as research use only targets for protein production. And so we start on the left at the project design level, where we really begin with just the electronic sequence design, going through optimizing the gene ultimately to result in higher yields of protein production. And then we can actually do the DNA synthesis as part of that. And we will do the prototyping in-house and send you your gene of desire. And if you'd like, we can actually take it all the way through subcloning plasmid production as well as proteins. And at any step along the way, at any point in this timeline, you really do have the ability to choose what's relevant to your organization and your needs at a given time. You don't need to go through the entire process to work with our group. You can truly pick which aspects of this continuum that are most relevant for you at the time of project design. And so for us today, the items that are highlighted in red, gene optimization, gene synthesis, and proteins, are the ones that we're going to be focusing on to, in particular, talk about how you can move from R&D to production in the fastest way possible. First, just for a little bit of context, you have in the protein expression space, very popular host systems that are used for expression. And so you can do cell-free expression, which does not have any kind of living components as associated with it. You can do bacterial expression through E. coli, or you can do yeast expression, insect expression, or mammalian expression. And we'll talk about primarily the complex proteins that you can make via mammalian expression, and we will talk about how we view that within our organization to ultimately provide you with the highest quality proteins developed from genes in the fastest amount of time. So as I mentioned, starting out with the mammalian cell lines that are typically used for protein expression, particularly in therapeutics development, you have two that are the most common here, the HEC293, or the human embryonic kidney cells, and CHO cells, the Chinese hamster ovary cells. And the benefits of mammalian cell lines is that you have the ability to really do post-translational modifications as it compares to using another type of expression system. And that's really important when you're thinking about developing proteins like monoclonal antibodies or bispecific or trispecific applications, because the reality is the mammalian cell lines are the way to go in terms of getting the highest yield in the fastest amount of time at the highest quality. And different cells, whether it's the HEC293 or CHO cells, have different uses at different times, and we have that flexibility within our organization to be able to, one, not only offer both, but two, as an outsourced service, we will also make recommendations about which ones to go through for that optimal protein expression. And so in terms of transfection strategies, we have a couple of options that you can use here within our GeneArt services. And GeneArt is the name of our organization who really focuses on that R&D range of protein expression from gene to protein. And so our approach to it from a strategic perspective is to have transient expression for 4 to 14 days, which is how long it can ultimately take depending on the sequence that you are trying to design and then ultimately express. You go through a high efficiency transfection as part of the process, through expression, and then ultimately protein production. So that's very fast, right? And we're trying to talk about the difference here between what is transient expression compared to what is stable expression. Stable expression really takes months in a lot of cases. You're trying to get a stable cell line at the end of your process so that you can ultimately, in a reproducible and scalable way, make the protein that you're trying to make as your target on various scales and timelines at different times of the project that you have in mind. So to kind of summarize, the way that I like to think about transient expression versus stable expression is that transient expression is really when you have a lot of squishiness or a lot of uncertainty around kind of what your gene sequence is and how it's going to express when it gets into a host cell system. And then after that, if you find a candidate that's kind of working, or you find a sequence and ultimately clone and then plasmid that works for you and works at various scales, because at GeneArt we can scale up from 24 well plates all the way up to 25 liter production scales. At the higher end, you really do start to get a greater sense of, okay, here's what stable cell line development would look like. And that can take months. And this really truly isn't comparing the two. It really is that transient expression, in a way, from our perspective, precedes that stable cell line development. And the fastest way to do it when working with our group, GeneArt Services, is to go through our outsourced transient expression workflow, which can be as quick as 4 days or as long as 14 days, depending on the sequence and the approach to actually doing the expression. So to kind of summarize, at the very beginning, GeneArt will be there. for most of the uncertainty. That's where we specialize, is when you don't know what's going to happen, call gene art. That is definitely where we excel in that transient protein expression space. Now, focusing on the discovery workflow here, we're going to start at the very beginning, right? Going from project design through gene optimization and then ultimately to proteins at the very end of the process. But let's start right at the very beginning. When you are starting off, we at GeneArt will ask you to send us a sequence associated with what you are trying to ultimately express. We have a proprietary patented algorithm known as Gene Optimizer. It's our technology that really takes into account many parameters to optimize the mammalian coding region. And this allows us to optimize expression to result in the highest yields that you can get right now today. We're very excited about this unique process that we have to optimizing gene sequences. And when coupled with our production processes, we've seen very strong increases in terms of yield resulting from the expression that we do following the optimization. And to talk a bit about really just our world-class performance in this regard, we have a couple of studies that have been published and we can get access to them. And you can actually just look them up as well. They're available, but if you need a link, feel free to reach out to your Thermo Fisher partner to be able to receive a copy. We have basically compared within this study kinase protein expression. And our Gene Optimizer software consistently provided higher expression levels compared to the wild type gene, as high as 200% and as low as 25% expression. We have a very high reliability with this algorithm. It's very unique. We have some really talented folks within our R&D organization who worked very, very hard at the molecular level to really understand what makes sense when you're building an algorithm to optimize that protein expression. So for those proteins that were tested in this study, the technology did provide the best results out of the different studies, and no other optimization algorithm, and that's what you can see in the 1, 2, 3, 4, 5 in each of the studies, showed consistent results. And you can see in the paper here, the GeneArt sections, just exactly how much the optimization, how much of an impact the optimization actually had. And it truly does outperform all other algorithms on the market today. This is a selected slide from another study that we posted as well, or we published as well, where we looked at different protein expression types. So the last one focused primarily on kinase proteins, but this really is a study that was done very in depth to demonstrate that different types of proteins also benefit from being optimized through the Gene Optimizer, whether it's transcription factors to ribosomal proteins, to membrane proteins, to cytokines. You can see the amount of increase that the optimized gene resulted in compared to the wild type. And it's pretty impressive, even in situations when you are, let's say, not with the wild type getting any expression at all. If you look at the bottom one, the cytokines, in that particular case, the IL-2 only expressed in the event of the optimization in terms of relative expression. And that's just a testament to how powerful the Gene Optimizer algorithm truly is within the GeneArt environment. And then, as I mentioned, when it's coupled with our production processes, you're truly getting the highest chance of the best yield that you can get at the highest quality as well. I'd like to move into talking about the synthesis and expression part of our presentation. Again, staying within the gene art family within Thermo Fisher Scientific, we're moving on to really that next step, the actual synthesis itself, now that your genes have been optimized. So we perform really next generation gene synthesis within our Thermo Fisher Scientific gene art group. We, of course, as we mentioned, started out with gene optimization through basically bioinformatics and what today would be considered machine learning processes. You move into oligosynthesis, then gene assembly, then cloning, sequencing, and final documentation. Everything I just went over really truly is a sequential process, and the process that we follow within our group to make sure that you're ending up with not just a cloned gene, but we're really verifying it. We're making sure that you are getting increased reliability, even with a shorter turnaround time that we're able to achieve through our processes in-house. We are able to do very high throughput. That's something that's fairly impressive, at least from my perspective, in terms of our production partners at our manufacturing site. They're very fast. They've been doing this for 20 years, and we have a really excellent team who not only takes the time to do the production, but will also work to the point that they can troubleshoot things. We do protein production all day, every day, and that's a unique thing about this team. They're very, very experienced, and they are able to really go from that optimized algorithm to the expressed gene, and then, to the gene synthesis, and then the clone gene, and making sure that if there are any troubles along the way, that we will do our due diligence to make sure that you are getting the highest sequence accuracy possible. And so all of these things combined, doing all of that in-house, especially starting with the gene synthesis itself, it really allows you to go, okay, ultimately at the end of my project, I'm trying to get an express protein, antibody, whatever it is. And you start to wonder, okay, well, how can I increase my chances for the highest yield possible? And that's what we mean by next generation gene synthesis from our perspective, is really starting at the electronic level, or the digital, from a digital perspective, where you optimize the sequence before even starting with any of the biology. And then after that gene synthesis, you have the ability to move into bioproduction with a bit more confidence. And so after the gene synthesis, you can really move into an expression plasmid and then do actual protein expression as well. You can see in the box that has a square around it right now. These are really the next steps in that process. On our way toward protein production, you will go from that clone gene to an expression plasmid before ultimately doing the expression. As we mentioned before, we really champion that gene optimizer algorithm to increase your protein yield from your expression plasmid. We have a lot of design flexibility in how you want to do your plasmid preparation, which is very high quality, and we do it all in-house. And that's the great thing about working with the gene art group within Thermo Fisher Scientific, is because you're starting your journey with the same group all the way up to getting that protein expression step done. Along the way, you can have full control over how your process is going. So starting from the sequence, like I had already mentioned, all the way through at this point, the expression plasmid, or the plasmid preparation, gives you that ability to have uniquely full control, as opposed to if you were to send in your clone genes to another provider, which you may not have control over the different steps of the process, one unique thing about the GeneArt group is because we do all of these services in-house for our partners, that provides the unique ability to control the different aspects of the process as you progress through it. And then moving into expression, we do have a number of flexible expression scales. As I mentioned, you can go as small as 96 well plates for some of our more high throughput or small scale protein expression services. A lot of our partners will start in the 100 to 300 milliliter range in terms of their expression after the transfection. And as I mentioned, when you're looking toward therapeutic and vaccine development, a lot of the time we're going to be recommending, or you're going to be coming to us with those mammalian expression systems, which in this case, we really prefer the XP293 and XP-CHO. But we will also do insect expression, because there are certain proteins that do express better within those SF9 insect cells. And so that's something just to be aware of, that we offer it. But truly where we excel is in that transient protein expression of those mammalian expression systems. For the right part of the slide, I'll let you read it. I'm going to get into more detail here in a second, but our approach overall, if you look at the bottom line there, our approach to bioproduction is really collaborative. We want to be a strategic partner for you. We have a project management team who will help you with getting up to speed. Our local manufacturing team is also co-located with some of our project management team and some of our sales support and customer support staff. And so that benefit of doing all of it in-house with GeneArt is one great way that you can really drive that performance through working with us through that entire process. And so for the last part of this presentation, and I really do appreciate all of your time today, we are going to talk about how we ultimately increase protein yield through the actual expression and purification processes that we offer within Thermo Fisher GeneArt. As I mentioned, there's a lot of advantages to using the XP293 and XP-CHO mammalian systems that really express more protein per cell. That's what everyone's trying to get to. How can I get the highest yield for in the fastest amount of time for the lowest amount of cost? Using these expression systems is a great way to do that. And in our case, you can see in that first red box there, using these expression systems will allow you to have a higher cell density, meaning on a per cell basis, you're going to have higher productivity and a higher efficiency transfaction as well. And you can see that the protocol is very easy. It plugs right into the 5 to 7 day workflow that usually goes on when it comes to using the expression systems. But what I want to highlight is when paired with our gene and vector optimization, as I mentioned, more often than not, we are seeing a 3 to 10x yield improvement by combining all of these factors. And so you may, in certain cases, see those benefits on the right, or on the left, but they are enhanced when you combine them with that gene optimization that I mentioned earlier. And it's more reliable and results in a higher yield than you would otherwise get without doing the gene optimization. And as I mentioned, I really encourage you to take a look at those studies I highlighted earlier. Just a quick bit more explanation in terms of the Gibco protein expression systems. They're industry leading. It's likely many of the participants of this call are very familiar with them. In terms of antibody screening, immunogen production, membrane and intracellular protein expression, On the left, you're going to see HEC-293, the cell-based system that can have a protein yield up to 1 gram per liter in terms of yield. And on the right, XP-CHO, really, as far as CHO cells are concerned, another industry-leading cell-based system, you're going to be getting protein yields up to 3 grams per liter, which in the bioproduction community really is toward the upper limit of capability, especially if you're getting them on a consistent basis like you would be within working with the GeneArt framework. A lot of the applications for CHO are some of those more advanced, newer applications like bispecifics, other multi-specifics, and then of course antibodies are really, monoclonal antibodies with CHO cells. Those are really popularized workflows that I'm sure many of you on the call are well aware of and experienced with today. And just to highlight a bit about the XP-CHO system in terms of the expression levels, the titer in terms of grams per liter, you can see the comparison versus the Freestyle CHO, which is another CHO system, the XP-293, and then ultimately the XP-CHO system. And you'll see just a massive increase in terms of expression levels comparatively with this XP-CHO system. And so it's definitely worth using. We love it in-house at GeneArt. We do recommend it for a lot of our mammalian protein expression projects, and we're very interested in helping to identify exactly why this particular cell expression system might be best for your sequence. But again, one of the benefits of working with GeneArt is with all that experience, we can do the exact opposite. We can also tell you when not to use it and when there may be a better alternative as well. But more often than not, this XP-CHO system, we're very, very big fans of. We highly recommend it. And that's something that we're really, really happy to be able to use in-house within the Thermo Fisher family of companies. So moving on to the final parts in the R&D scale bioproduction, you're going to be interested in purification and analysis ultimately. We have a number of ways that we can do purification. We do a lot of them, as you can see inside that cell. Inside that box there, we're very experienced in this, and we feel very comfortable with it. And in terms of our analysis, we have a number of ways. Some of our standard processes are the SDS page, nanodrop, capillary gel electrophoresis, but we can do some other approaches as well. HPLC when it comes to size exclusion chromatography. Also, we can and have done with a lot of our partners endotoxin detection. That is a process that we do get requested a lot, and it's very important in particular when you're dealing with, for example, in certain cases, toxic sequences, which can sometimes happen, but are significantly lower risk when you're starting with that gene optimization process that I mentioned earlier. So just talking a bit about our gene to protein services, the purification approaches, you can see everything in the box there. We've got a number of different chromatography approaches that you can take to purification. We also have different types of affinity tags, but we also do provide unique customization options, which is pretty unique, I would say, to a provider like Thermo Fisher, who does do a lot of proteins, oftentimes on a high throughput basis. We will do everything from concentration adjustments all the way to things that aren't listed here, like choosing custom tubes, for example. You let us know what your needs are, and we will do our best to absolutely help you toward making your project go as smoothly as possible and as quickly as possible. And as I mentioned, we have a lot of different ways. The filtration and purification approaches that we have are not just one one size fits all, so to speak. We understand that every project is unique, and I would go so far as to say we understand it better because a lot of times our partners start at that gene optimization level, so we can see it coming in our production pipeline and really understand when we may or may not run into purification challenges. So that's another benefit of working for your entire production workflow at this early discovery stage with our gene art services. Here's an example, and this is my second to last slide here, of some of the documentation that you might receive early on working with our gene art services here. We will provide a certificate of analysis, results of the chromatography, for example. We really want to make sure that you have a lot of confidence in trusting us as your outsourced provider for transient protein expression, for those early services that you might be looking at. You know, it's one thing to be able to do it in-house and feel like you have control over every step of the process, but we understand that for some people on this call, that can be cost prohibitive from an equipment perspective or an investment perspective. And so we want to make working with gene art really feel like an extension of your laboratory. We want it to be personalized to you so that we build that trust over time and really can grow with you. We want to help scale with you. We, value every single one of our customers. It's not that we will just push your proteins through as quickly as possible. We want to treat every single customer that comes to us with the respect that we would have doing all of our projects with our own R&D team. And our manufacturing team, with all of their experience, they truly have seen it all. And I can tell you that we're very proud to talk about how our team cares so much each and every day about the projects that they work on. And finally, I'd like to just share a little bit of information about upcoming services, and we actually do provide this on a pilot basis right now, the high throughput ex vivo expression and purification service. In as fast as 21 days, you can go from sequence all the way through purified protein with that analytical report in either 24 or 96 well plate. That's really great for antibody screening. That's something that we have a lot of partners, a lot of the bigger partners that you are probably very familiar with, like this service, because it allows them to explore a number of targets all at once before ultimately scaling up to higher scales or production volumes. And so, you can save a lot of time, a lot of money. We understand things are getting smaller. We understand that needs are getting faster, but maintaining the higher quality product that you're used to. And we can say that as we scale down, we are not sacrificing quality by any means. And this is one new product offering that we're very excited to have and offer you today.
We now ask that you participate by answering these poll questions. Our first poll question is, would you like to receive a quote for an early stage protein production project? Remember to record your answer, click on your selection. Again, would you like to receive a quote for an early stage protein production project? We'll move to our next poll question. Would you like to be contacted by a technical sales specialist to understand more about our protein services? Please select yes or no. You record your selection by clicking on the answer. Would you like to be contacted by a technical sales specialist to understand more about our protein services? And then our final poll question, if you have a de novo gene synthesis project currently in process, either in-house or outsourced to a provider, when do you anticipate you'll require protein production services? 1 to 3 months, 2 to 6 months, or 6+ months? Click on the answer to record your selection. If you have a de novo gene synthesis project currently in process, either in-house or outsourced to a provider, when do you anticipate you'll require protein production services? 1 to 3 months, 2 to 6 months, or 6+ months? I'll give you a few more seconds to record your answer. Thank you for participating in the poll questions. We will now be hearing from Dr. Jeff Howe, representing the Pharma Services Group within Thermo Fisher Scientific. One of the goals today, and the reason for doing this webinar, is to explain how you can go from the R&D environment to commercial scale production. At GeneArt, they are proud to take on the earliest stages of your research, even when you are not sure what is going to happen, all the way to the point when you feel comfortable moving to mouse studies and ultimately clinical trials. By working with Thermo Fisher Scientific, you get the benefit of that early stage transient protein expression, the discovery services, and then how to move quickly into clinic and ultimately to commercial scale production in one seamless, integrated way within the Thermo Fisher framework. Jeff, you may now begin your presentation.
Appreciate the introduction there. So I hope everyone's doing well, and let's kind of move on from the R&D and into the production of the bioproduction side. Just want to refocus the audience here a little bit. As Steve kind of presented, going from concept to commercialization is quite a big endeavor, and I just want to try to break this down a little bit further on the next slide as you move out of discovery and into development and subsequent stages. So Thermo Fisher Scientific will continue to be your partner as you move out of that phase. And it's really important that we overcome, or once you've overcome some of those development hurdles, as presented by Steve, we have the offerings that are necessary to get you through the next stages. Now, for the rest of this presentation, I'll focus on the development section. I will refer to some of the clinical and commercial aspects, but I'll focus on some of the development elements in the rest of the slide deck. So really the key is to really leverage the experience of a partner that can offer a really mature platform which is built that allows for easy transition into a predictable supply chain so that you are getting the product if and when you need it. So this development process, these solutions that we're looking for to create this new therapeutic is really critical first step in moving into some of the clinical trials and commercial production. So our capabilities truly demonstrate an end-to-end service offering from DNA to patient, and that's really bounded by the foundations of quality. So if I was to kind of now focus in a little bit on the process development optimization side, once you come out of that discovery piece, we're really focused on the process development elements of your molecule. So that includes things like cell line development, cell culture customization. There could be certain analytical aspects that we want to look at, whether it's related to characterization or some post-translational modifications, or it could be to do with scale-up or tech transfer and in-process monitoring. as well as finally, generating some material, whether it's for that tox study or reference standard. And these are really important factors as we now embark on this journey of bioproduction and move into both your clinical trials and hopefully into that commercial launch. At Thermo Fisher Scientific, specifically within the pharma services, we have these world-class product capabilities to really drive you there. These capabilities are definitely industry leading and end-to-end. And what I mean by that is that we have been extremely patient-centric in delivering over 160 molecules, over 20 years of manufacturing experience, really compressed into some of the capabilities and service offerings that we have to offer to you as our customers and ultimately to our patients. It's truly end-to-end from development to manufacturing for both DS and DP, as you can see on this right-hand side, moving from both material packaging, distribution, and ultimately logistics for both clinical and commercial. And that has really paved the way for a lot of success for some of the molecules that we have developed for our customers. So you're not alone when it comes to evolving the R&D pipelines on biologics. Here we see some market projections as well as how we've gotten to this point. With a CAGR of 11.4%, right? This pipeline, continues to be extremely strong, even during COVID times. Now, while there is a wide diversity of molecule types, almost 40% in that preclinical phase revolves around monoclonal antibodies, and that's definitely one area that I want to try to help target today. It is around these monoclonal antibodies. When we discuss development, the monoclonal antibodies certainly provide some baseline on timeline and speed, but with changes in molecular complexity, as you can see here on the right-hand side, whether it's things like ADCs or fusion proteins, bispecific antibodies, we can use that platform that we've developed for monoclonal antibodies as a basis to further evolve the scopes so that we can explore greater design spaces as a way to de-risk your CMC activity. So even though this presentation will focus specifically on the development and MAB side of things, I think the other 60% we can certainly tackle with you as well, based on, again, the capabilities and experience that we have gathered throughout our 20 years of experience. All right. So then, now that let me pull you back to what we have discussed so far, which is now that you've started your engines on discovery and working with GNUT to generate those first pieces of asset on the construct and on the molecule specifically, your next challenges will likely be on that first in human. How quickly can you get into first in human and the speed into the clinic, as well as delivering a robust and productive process into manufacturing. So as you move along this path, as you get into clinical and commercial, there's a certain demand that is required, and certainly ensuring that you have a process to meet those demands is going to be critical. This now puts the spotlight on things like cell line development, process development, and potentially that scale-up pilot scale as paths that will now fall on the critical timeline if you're thinking about speed to clinic. All right. So then, speed can be formulated in a number of ways. At Thermo Fisher Scientific, our service offerings are being modernized with the latest technology and automation. Throughout the various functions of development, we have invested in advancements in developing new technology and automation that will take us there faster. So, for example, if we look at some of the automation and latest technologies that we have invested in, we start with cell line development, a really targeted approach utilizing microfluidics and OEP technology for single cell cloning. Moving into some of the process side, where we have the AMBER 15, as well as TCAM robotics to really facilitate both the upstream and downstream process development at the smaller scale. And then the process and the cell line, I should say, is complemented by analytical and characterization foundations through the multi-attribute method, and that's using a workflow that leverages LC-MS methodology to obtain product quality attributes such as glycans and charge variant data to now really shine a light on product quality as you move into bioproduction. If I was to kind of take one example, let's focus back on the Beacon platform. As mentioned, the Beacon platform is a microfluidic system that uses OEP technology, so optoelectrode positioning, and it uses light to help us with single cell cloning. Single cell cloning traditionally is done through limiting dilution, but with the Beacon, we're able to now able to seed cells into a microfluidic device and then sort the cells based on monoclonality. If you look at the schematic on the bottom, the instrument is on the left with the image in the middle showing how we're using OEP to channel cells into what's called these nanopens, so channels within the microfluidic device, and then we're able to assess both growth and productivity over a 5-day period. So this has introduced significant amount of automation as well as modernization to the way we deal with cell line development and to the way we introduce a cell line into the bioproduction pathway. So realizing speed to IND or IMPD is not just accelerating the platform. The quick to clinic offering is our offering which has gone through careful construction and integration of a number of things. Certainly the first one is around the workflows. It's around the Beacon, it's around the AMBER systems and the miniaturization that we're able to get out of that workflows, as well as the analytical methods. So as we develop the molecule and the process, there's understanding of the process that's required through various analytical methods. But I think what's more important is the robustness of that platform. With over 80 PD programs since 2017, and now a revamped XP CHOS cell line, which I'll speak to a little bit more detail in subsequent slides, is really getting us to the productivity that we're looking for, and then also leveraging the process development experience that we've had through those programs. And this is then combined with assessing risk, and you'll hear me talk about risk throughout this presentation, which is really critical as we're thinking about moving to that IND or IMPD. So we talk about timelines, we talk about accelerating, we talk about speed. So this is our quick to clinic 2.0 timeline. It's an accelerated and optimized start to finish workflow that, as you can see, draws on a number of parallel activities. We have taken a really careful approach to identifying what's on the critical timeline and then optimizing the workflow accordingly. And this is the end product of that approach. You can see that some assumptions are made through the workflow, but what's truly impressive is that this is not just completing a GMP batch at the end. It's integrating the PD to the tech transfer as well as the GMP operations. And finally, we're able to release your DS and DP within that time frame. And this is all because we are with you every step of the way to really get you closer to that IND or IMPD submission. So moving along then, having seen the timeline, I'm sure by now you're wondering how are we addressing some of the risks and with the amount of concurrent work. Mitigating predictable risk is certainly embedded in this offering, in the quick to clinic offering. And there are three key pieces. Our approach is really leveraging a well-characterized cell line, which is coming up next. That will help reduce inherent risks and failure points as we move from discovery to bioproduction. And then it's about advancing our platforms. So we have invested heavily into developing this platform that really is able to pull together the critical pieces that you need in getting you to that IND. So then Quick to Clinic 2.0 has definitely been focused on a revamped cell line, and that being the Gibco Freedom XP-CoS cell line. So Steve had mentioned, some of the work that has been done through his group, and more importantly, the HEC-293 cell line as well, the XP-CoS, being really critical foundations in the R&D environment in generating some of these products, and now we've really turned the transient system into a stable one. Certainly for our workflow at the very beginning, where we're doing cloning and gene synthesis and some of the plasma prep work, that is supported through our GeneArt entity. And really, this is our transition point where we're going from discovery into bioproduction. What's also really critical here is that anything that you learn through discovery is maintained, right? Whether it's through vector design or other things, those are all maintained as we transition to bioproduction. We don't want to lose any of the really critical aspects that you put into the design of your construct through this initial process. So this is really the handshake that you see going from GeneArt into the pharma services. So when we think about the Freedom XP-CoS cell line, it's the same robust cell line, but certainly GMP banked that you've used through the transient system. It has been adapted for high density culture, getting to 2x7 to around 2x7 or 2x10⁷ cells per mil. It's been documented in literature of its rapid doubling times, as well as the ability to produce a high amount of protein. As you can see on your right-hand side, definitely in the grams range between that 3 to 4 range. And then when we think about the cell line and the stability of the cell line, we've seen good stability across many of the clones that we've generated, whether it's for MAB or recombinant protein. And certainly we've seen really high quality and very active protein where we're looking through some of the analytical methods. So if you take one message away from this slide, it's to say that the cell line, the host cell line that you're using for the transient, your transient operations will be the same cell line that you use for your stables. Now, consistent quality between transient and stable clones is really critical, and I think that As Steve had mentioned, certainly for the XPCHOS system in the transient format, we've seen gram ranges as demonstrated on the left-hand side. We've also seen very comparable product quality. Here you see some N-glycan data moving from transients to stables with a 400 times increase in scale as well. And certainly, while we see some differences, the G0F still remains relatively consistent throughout. And the final piece to the Freedom XPCHOS is the regulatory support information. There is a master file for this particular cell line in the form of a regulatory package that provides the information around the cell line, the history, the lineage, as well as characterization. There is certainly an application data package that's available, and then the lineage, as mentioned, in supporting your regulatory filing. So just a couple of slides left. Now going through and introducing to you the stable system, the stable XPCHOS system, and how it fits within Quick to Clinic 2.0. We've really laid the foundations for success for your IND or IMPD. So hopefully, you can see that Quick to Clinic is formulating the pieces that are really critical for this foundation, where it combines the process, the analytics, as well as the scale-up strategy with a really robust cell line that is available now to create your molecule of interest. It's certainly through some of the slides, you could see that it's been integrated into our platform. It continues to be royalty-free or exit fee-free, and we have a significant body of data around scale-up solutions well beyond your IND and IMPD milestones. To kind of sum my section up, Quick to Clinic is just that first step, and really getting your molecule past the IND and IMPD is that first step in the drug life cycle. I want to highlight here that certainly Pharma Services is able to help you go beyond that, going from both lab to clinic and then to commercial. So we have some additional services which I'm sure that there will be opportunities in the future to kind of share with you. So let me sum it up, and I'll try to sum up Steve's section as well if I can. So as Steve presented, partnering with Thermo Fisher Scientific can significantly speed up your development timelines. Starting with gene synthesis is the fastest way to screen and to screen those clones to identify the candidates that's necessary for that next step of bioproduction. GeneR services, through what Steve had presented, can accelerate the protein R&D timelines and really simplify your process. And I think ultimately what I took out of what Steve had said is really help you come up with a solution in addressing some of the challenges as you go through that kind of workflow. Now, while the speed to clinic is one of the most critical goals coming out of discovery, it shouldn't be prioritized at the expense of other critical success factors. And I hope that that risk-based assessment that you saw throughout the presentation helps to address that point. And then work with things like Thermo Fisher Scientific, who can offer seamless scale-up solutions beyond the IND and IMPD milestone. Certainly, this is really critical, and we should kind of ensure that there is complete end-to-end for any molecule or any drug that you're trying to funnel through that life cycle approach. And finally, a quick-to-clinic program for monoclonal antibodies that's designed to take you from transfection to IND in as little as 13 months. It's certainly market-leading, and we balance the risks to the reward as well. So with that, I'll move over to that final slide. Just some contact information or any additional information that you need, please reach out. And with that, I want to thank you for your attendance to this webinar.
Thank you, Steve and Jeff, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is for Steve. Can we ship clones from another provider for gene art to perform protein expression and purification projects?
That's a great question. The answer is yes, you absolutely can do that. One of the things that we do encourage within gene art services at Thermo Fisher Scientific is the fact that we can do the entire continuum from DNA sequence all the way through expressed and purified protein in-house. So you will get a higher level of service and likely higher level of quality as well by working with us at Thermo Fisher Gene Art from the very beginning. And one thing that we can do as a result of doing that is at any point in the process, you have the ability to have better control over everything. Sometimes there's a risk in shipping clones in that there can be, mislabeled tubes, there can be shipping conditions or packaging that are not up to par that we don't have any control over if we receive it in that way. And if the reason for wanting to do that revolves around pricing, we're more than happy to discuss options to really provide you everything that you need in-house from that sequence design all the way through the purified protein. So feel free to contact one of your Thermo Fisher partners who you work with, and we'd be more than happy to discuss the different options available to you.
Thank you. And it looks like we have time for one more question. This question is for Jeff. How are you addressing current challenges around raw material sourcing with regards to the quick-to-clinic solution?
Thanks, Marie. Yeah, no doubt this one's on everyone's mind. It's certainly a challenging environment right now. That said, with this kind of platform approach that we're taking, there's certainly a standard set of single-use components and a standard set of resins and media that we would use. And we're able to really procure those well in advance in preparation for the GMP manufacturing batch. And sometimes even before, prior to that signature. So really it depends on where we are in terms of the discussions with contractual items and things like that. But there's always, with such a changing environment, there's always opportunities to go through and identify the critical timeline items that's stopping us from moving forward. So and we're always open for that discussion. So that will be my answer to that, Marie. Thank you.
Thank you, Steve and Jeff, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsors, Thermo Fisher Scientific, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until April 21st, 2024. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.